is that not? Oh, now it's advancing. So my lab is using mouse model to study how muscle stem cells or satellite cells are maintained for a lifetime. Michael Webster, a former postdoc, did the intravital imaging to show that the remnant of the dead myofiber, so-called the ghost fiber, serve the guide for myogenic progenitors for proportional regeneration. So then Michelle Rosso and Patrick Lee, two former students, then are very interested in what does the satellite cell recognize the ECM. By that, they study the integrins. So integrins are heterodimers forming with beta and alpha chain, and satellite cell unfortunately express many of them. In particularly, beta-1 and alpha-7 integrin form the receptor for laminin. So in their work, what they find is when you knock out beta-1 integrin just in the satellite cells, those muscles completely fail to regenerate. And the defect involve quiescence, proliferation, migration, differentiation, and fusion, meaning almost every part of it. And in collaboration with Florian Benzinger's lab, we show that these knockout beta-1 integrin satellite cells have real defect in binding to laminin, fibronectin, as well as collagen. And in our collaboration, we actually showed that further explore the role of fibronectin in muscle regeneration. However, before we actually published our work, there was another study showing that beta-3 integrins are essential for muscle regeneration in that using the beta-3 integrin germline mutant, there were almost no regeneration. So because beta-3 alpha 5 and beta-3 alpha V form fibronectin receptors, well, this really piques our interest. Unfortunately, beta-3 integrin germline mutant have a severe bleeding clotting defect. So when you use injury, that really complicates the explanation for the muscle injury regeneration phenotype. So to really explore whether beta-3 integrin have specific cell autonomous function in the muscle lineage, we generate the satellite-specific knockout. So using germline pre as well as conditional, the result is the same, that we do not see any muscle regeneration defect either in male or female. And this we done early or late time point, we do not see anything. So we would suspect then the previous data is because they couldn't clot their blood, and there somehow is a secondary consequence of that defect. Or alternatively, that beta-3 act in a non-muscle stem cells within the muscle that support regeneration. Whatever it is, if beta-3 does have a role in satellite cell autonomously, that role most likely is compensated by other integrins and presumably beta-1 integrin. So moving downwards, we're interested in other signaling components of the integrin signaling that may we can assign specifically for each part of the role controlled by the beta-1 integrin, such as quiescence, activation, proliferation, and so on. So fortunately or unfortunately, the FACT knockout has been reported by Tom Randall's group, and we can deduce from their work there are virtually no defect in the early stage other than the primary defects they can assign to fusion. And the IOK function in the muscle have been addressed, but within the satellite cell biology are largely derived from a C2C12 study. So we generate the conditional knockout, and we are satisfied to see it does have a muscle regeneration defect by histology, MH staining, as well as counting myofiber area, as well as fiber number. However, we were surprised there was absolutely no satellite cell number change or proliferative fraction, and we can carry out this in vitro. We do not see proliferation defect again, and even by looking at Pax7 MyoD positive cell fractions, we do not see the fate fraction change of the mutant as well. So then we went back and looked at the myogenic. So that's for the early differentiation or whatever. And we were surprised that even though we see a clear defect or reduction at 5-day DPI, at 3 days, there's virtually no difference. Then that would suggest to us whatever the cell loss or the defect happened in the terminal differentiation or fusion. To test that, we did, I hope you can see. So we did in vitro imaging of the satellite cell fusion. And you can see the control, they can flatten out, they move around, flatten out, then fuse. The mutant are much more round. They can move around, not a problem. And they can differentiate, to assure you they can differentiate just fine in vitro, but they really do have problem with fusion. And during this time, we don't see cell loss compared to in vivo. So the question remains, what are the signaling component of integrin that immediate early step of before fusion? And if FAC and ILK both mediate satellite cell fusion, do they use distinct mechanism or the same mechanism? As far as we know, we have checked the mechanism proposed for FAC mutant, and we do not see that with the ILK. For now, I'm going to quickly switch to the other side of the satellite cells. So a few years back, Robert Krause's lab done really beautiful work to show a collective of apical cadherins are important to maintain muscle stem cell quiescence, just like the beta-1 integrin. At the same time, we actually report actually the same issue of that journal, that there's another apical component called MPP7 protein. It is also localized to the quiescence satellite cell on the apical side. However, by RNAi and in vitro studies, we can only derive the conclusion that is involved in satellite cell renewal as well as proliferation. So we have since done the knockout studies, but before I go into it, I probably need to give you an introduction of what MPP7 is, because there are virtually no one studied them except us. So MPP7 stands for membrane palmitoylated protein 7. It has 3 membrane targeting domains. The first L27 domain binds to LG1 and Lin7, and it has been reported to target MPP7 to adherence junction. The third SH3 hook domain supposed to bind to PLS1 or MPP5 and crump and target them to tight junctions. And in that study, the RNAi data show it will knockdown of MPP7 affect tight junction, but does not affect adherence junction. And that leaves the middle PDZ domain, which is also required for membrane targeting of MPP7. No one knows what it binds. Because crumps are crumb 1, 2, or 3, none of them are expressed in satellite cells. So we sort of do not believe MPP7 function in the satellite cell through a classical tight junction. So for that reason, we sort of explore another possibility that has been proposed a long time ago, that MPP7 form a complex with angiomotin like 1, like 2, and MUPP1. And I know this is all very foreign to other people, and this is really never have been pursued. Or what they suggest that it is some kind of junctional regulator, but they did not assign what junction it will be. So from now on, I'll focus on MPP7 and angiomotin. So as it turned out, the PDZ domain of MPP7, that one is required for membrane targeting, happen to bind to angiomotin directly through angiomotin's C-terminal PDZ binding motif. This is done by co-IP studies using 293T overexpression system. And perhaps most pleasing to see is when we knock out MPP7 using Pax7cm-driven Itgb3 generated by Dr. Lepper. We can see both of the MPP7 as well as angiomotin knockout have almost similar phenotype in that they have reduced regenerative myofiber as well as smaller fiber. Furthermore, we can assign most of the phenotype or defect is due to satellite cell proliferation defect by not just a Pax7 number, but also a proliferated fraction of Pax7 positive cells. And that's, and we can trace it even up to 21 days. And for whatever the fiber we can find, the fiber is smaller and the Pax7 number is also reduced. And similar results are found in the angiomotin mutant. So we can also carry this as the famous single myofiber assay. And I hope you can appreciate that both MPP7 knockout as well as angiomotin knockout have reduced red Pax7 only fraction, as well as reduced Pax7 MyoD double positive yellow fraction, and increase, of course, the Pax7 MyoD positive differentiating action fraction. So using this as a platform, we perform I call a pseudo-complementation assay by reintroducing the cDNA of MPP7 and angiomotin back into their mutant background. And we're pleased to find the wild type of each of the counterpart can rescue the phenotype somewhat. However, the MPP7-PDZ domain deletion or the angiomotin-PDBM mutation cannot do any rescue whatsoever. Strongly argue that this interaction domain between the two are the functional part for them to function in vivo. So now I come back to what I told you before, that MPP7 is apically localized, and that's based on single myofiber staining at T0 when we just isolate them. But what I sort of skip is that 48 hours later, most of the satellite cells at this point presumably activated and actually have MPP7 in their nucleus. And similarly, this can be done by just plating purified satellite cell on the dish. And at T0, most of them, I don't think it's membrane, we'll just call it cytoplasmic. And 48 hours later, almost all cells have MPP7 in the nucleus. So because it's localized to the nucleus at the time of activation, we're very interested in whether it actually affects transcription. So another pleasing result we found is that there are not too many downregulated genes or upregulated genes in both of the mutant and T40A. But the differentially expressed gene between the two are almost identical. They are hugely overlapping. And we think whatever is difference is really just the knockout efficiency difference that sort of changed the threshold. Because it's only so few genes, gold term analysis really doesn't help us that much. It doesn't have tons of pathways. But the three we can find is very loosely transcriptional regulation, GTPase activity, as well as the YAP and TAS pathway. And suffice to say, YAP and TAS mRNA level itself are not changed. It's just their downstream gene. So that really piques our interest because the implication of tapping YAP and TAS. And also what I didn't mention before is angiomotin actually bind to YAP and TAS directly through a totally different domain, not the PDC binding domain. So we are continuing to work on this direction. I'm going to disappoint you that we don't know the mechanism yet. But what I can give you a teaser is that indeed we look at the MPP7 knockout cell. We do see that initially the YAP and TAS at T0, there are no difference. YAP is in the mostly cytoplasmic. 48 hours, they go into the nucleus. In the mutant, we see about 50 to 70-fold reduction of the YAP nucleus signal. And perhaps most surprising is the TAS. We've seen no nuclear TAS in the MPP7 knockout, while the control had tons of TAS, and that presumably because both transcription upregulation reported by Zemet a long time ago.
5 minutes left.
Yeah, this is my last slide. So with that, I'm going to wrap it up. So what I told you is that basically both basal as well as apical contact are essential to maintain muscle stem cell quiescence. One can then imagine when you have damaged myofiber, the apical engagement is derailed. And we assume then that release the cadherins sequester beta-catenin into the nucleus that's proposed by and shown by Rob Krause. And we think somehow that also trigger the YAP-TAS nucleus shuttling that requires MPP7. So the other way you can look at it is only after fully regeneration from the damaged situation that you can reestablish the basal and apical contact. And we assume, other than all the signaling molecule, these two are the mechanical sensors as the feedback to signal that muscle integrity has been restored to stop cell-less activity and therefore maintain, to restore the proper tissue integrity and size. And with that, today I talked about Natalie as well as Anwen's work, and these are large, they're all unpublished. Hopefully we'll publish it this year. And of course, this is not possible without previous fantastic postdoc and students, and certainly not without the wonderful collaborator we have over the years. And of course, the funding agency. And I'll take questions. Hopefully I have enough time.
Thank
Any
you.
Questions? I can probably.
I have got my hands raised, so I'll jump in while other people.
Sure, Sherry, I can always.
Beautiful, Fan. That's lovely work, and the quality of the data is fantastic. So you showed MMP7 this dramatic downregulation of TAS, but not so much of YAP. A little bit less, yeah. So what about AMOT? Because AMOT cooperates with.
We think it is the Wells proposed the right thing. That is the whole complex. So we look at MPP7 mutant. We don't see the AMOT protein. We look at AMOT mutant. There's no MPP7 protein. That's why they have another phenotype. So we really think the original proteomic study was correct. It is the 5 protein. What I didn't tell you is we actually map all the interaction between the 5 protein. We know exactly how they interact.
So in the cardiac lineage, it's been shown that AMOT becomes nuclear translocated with YAP and affects cardiac proliferation.
Yeah. So we do think we don't know why here we need MPP7, and I'm sure Peter probably have different views. What I think I'm most surprised is in the previous slide, sort of not mentioning it, is in the literature, IOK in fact supposed to regulate, right? The basal component is one of the most important regulator of YAP and TAS. We don't know why here it is MPP7. But I do think that's what we see a little bit YAP. It's because we played on matrigel, it has a stiff substrate. So I'm not surprised we see YAP in vitro. I'm not so sure in vivo yet. But I want to tell you that single myofiber, they're still going. Okay. Just less. And you will say, why would that, based on the previous publication, would that be enough to have the mutant phenotype? And I'm sure you know that the double mutant of YAP and TAS does not regenerate.
Right. So it'd be worthwhile, because you're releasing tension of the fiber when you're putting single fibers in culture. It'll be worthwhile if you can do longitudinal sections on the muscle fibers in vivo to have a look at that.
You don't need to do that. I mean, bottom line is, we can probably just do it in vivo to see. The YAP and TAS NTR is pretty good. We just haven't gone to the in vivo yet.
Okay.
Oh,
thanks again.
We did do the RNA-seq with the TAP and YAS, T38. It is like 1,000 some genes are differentially regulated. So clearly YAP and TAS has more than just MPP7. But I think again, it's because we still have a little bit YAP going in, and that's why there's slightly different phenotype.